Good afternoon, ladies and gentlemen. We are pleased to present you the investigation of epithelial cells-inspired surface deformation mechanism with its design applications. This project started from a research question, which is how do cells build and differentiate in nature? Specifically, we looked into epithelial cells since epithelial cells are one of the most critical cells in the earliest stages of all living beings. Epithelial cells deform and proliferate themselves into multicellular tissues during embryonic development. These tissues will eventually become various organs and blood vessels in our bodies. They also form the skin of the creature, establishing a barrier for the creature's body from the external environment. We focused on the living mechanisms within these cells that facilitate tissue curvature in the organs, skin, and blood vessels, as epithelial cells are capable of reorganizing themselves into different shapes to envelop cavities. This capability allows epithelial tissue to establish barriers for the creature's body from the external environment and deform the barriers to fit with the environment. In this project, there are two parallel goals. First, we aim to investigate the deformation mechanism of epithelial cells to enrich our overall understanding of how the epithelial cells present the scutoid shape. By setting up a series of the parametric toolkits from the perspective of design research instead of cell biology. Specifically, this project looks into dialectical relations between global morphology and individual units through computational simulation and material study. Meanwhile, situating this from a design perspective, the paper aims to adapt the deformation mechanism of epithelial tissues and the concept of newly discovered scutoid into a novel design application. Notably, inspired by scutoid geometry, we designed a new type of brick termed Scutoid bricks. The bricks can be used to enhance the joint system in a masonry structure. As we surprisingly found that scutoid shape in epithelial tissue exhibits certain impressive features that we can potentially enlarge to a macro scale. Due to the limitation of imaging technology at the nanoscale, a comprehensive visualized description of epithelial cells' three-dimensional appearance has been missing from the field until recently. Most biological researchers understood the shape to be similar to columnar prisms or frustum shape. In 2018, through the approach of mathematical modeling, a group of scientists unexpectedly predicted that as the curvature of these epithelial tissues increases, some of the epithelial cells would likely develop into forms other than columnar prisms and frustum shapes. The research claims that this unique three-dimensional geometry is a transition volume between a pentagon and a hexagon with an added vertex. Researchers named the shape scutoid because of the backs of some insects, having a similar mini-triangular shell. The discovery of the scutoid has brought new geometric inspiration to understand the three-dimensional structure of epithelial tissues. Our research is fundamentally built upon three parts. We investigated the deformation mechanism through literature review and computational simulation. We then resulted in a scutoid embedded masonry shell as our primary design application. Beyond that, we conducted a set of material studies to bridge the gap between theory and practice. Studies found that the deformation capability of epithelial tissue is achieved through its cell rearrangement. Cells are moving around within tissue so that the overall morphology changes over time. As the primary mechanism of tissue deformation, epithelial cells rearrange themselves to a new position without being separated from the adjacent cell according to a topological T1 transition. A single layer of epithelial tissue is composed of adjacent and closely connected epithelial cells. When two cells engage with each other, the common boundary can be represented by a straight line. The length of this common boundary depends on the distance between the geometric centers of the two cells. When four cells are packed together, cells squeeze each other in both horizontal and vertical directions. If two of the four cells are neighboring with each other horizontally, the other two cells are isolated vertically. The distance of isolated and neighboring cells are inversely proportional. The closer the geometric centers of neighboring cells are, the further away the isolated cells are. Expanding this into 3D space, the deformation process of epithelial tissue is a surface that with thickness bends at a particular position. When the tissue is bending, the basal and apical surfaces associatively perform the T1 transition to rearrange its position. The basal and apical surfaces of any pair of adjacent cells that share one common boundary will have a different motion tendency. If the tissue is bent toward the basal surfaces of the two cells will two-dimensionally squeeze each other. At the same time, the apical surfaces will tend to separate from each other. In the process of tissue bending, with the increasing curvature of the tissue towards the basal surface, the boundary line between the two adjacent apical surfaces will gradually become shorter. The basal surfaces that two-dimensionally squeeze against each other will produce longer boundary lines. When 4 such cells are packed together, the two apical surfaces that get closer with each other will occupy the space that is vacated after the detachment of apical surfaces. As the tissue bends, the basal surfaces that squeeze each other will occupy the space left by another pair of basal surfaces. The discovery above allows us to establish a framework for building a parametric model of scutoid geometry. Our initial model is composed of 3 layers. Besides the 2 necessary outer layers to form the basal and apical surface, we also set up an intermediate layer to arrange the key vertex. The morphology of each cell in the cluster is determined by the length of the common boundary of adjacent hexagons. When the cluster model starts folding towards the basal surface, according to the T1 transition, 2 hexagons on the apical surface detach from each other. The common boundary gets shorter and the pentagons on the same layer merge closer. Conversely, the common boundary of adjacent hexagons on the basal surface becomes longer as hexagons are further apart, while the pentagons on the basal surface separate from each other. After reviewing related papers and establishing the parametric model for a single scutoid-shaped cell, we realized that the identification of scutoid shape should not be limited to the original definition, beyond the feature of transition volume from a pentagon to a hexagon. The number of polygon segments at the basal and apical surfaces of a prismatoid is an essential factor that determines the scutoid. We define 2 conditions that are required for prismatoid-like geometry to be recognized as scutoid. First, the numbers of the polygon segment are not equal on apical and basal surfaces. Second, 2 vertices on one surface can be merged into one vertex on the intermediate layer to equalize the polygon segment. These 2 new rules increase the possibility of applying the scutoid mechanism into design space, as the number of polygon segment is no longer a limitation. Based on the 2 rules we defined, even the prismatoid-like geometry that has 9 polygon segments on the apical surface and 6 polygon segments on the basal surface can still be recognized as scutoid. After establishing the parametric model of single scutoid and defining new rules for identifying scutoids, we looked into how to generate scutoids from a free-form surface, as in the biology world, scutoid can only be observed in highly curved space. Hence, we managed to develop a set of surface deformation generators that examines how the global morphology change will determine the deformation of a single cell. We invented an automatic workflow to form a 3-dimensional scutoid embedded surface subdivision and translated this into a C# code component in Grasshopper and Rhino environment. The generator starts with an initial input that changes the curvature of the surface. In this component, it is necessary to implement a simple associative data structure. It is a spring force model of a dynamic system combined with nodes and edges in this case. The surface is interpreted as a simple non-interacting particle system where each element is a simple point whose location changes over time, as a response to external forces applied to it obeying Newtonian physics. Some collections of objects can be updated in real time. The initial spring force structure contains multiple polygons that are optimized from the Voronoi diagram. The cells on the margin are fixed. Then, external forces are applied to each node in the spring structure, which makes it bend gradually. During this process, for cells that have edges and nodes that are overextended, a trigger function is added to generate scutoids. 2 vertices on outer surface merge into one vertex in the middle of cell. Hence, the scutoid generator becomes functional when the overall surface is about to lose the stability of the original form. Then, we started to think about if we can create scutoids beyond the digital simulation. What will be the best material to reflect the characteristic of cells and scutoid shape? We reviewed several papers and found that in the physical world, when cells are rearranging themselves to a new location, there are 2 geometric constraints that need to be considered, which are expansion force from the cytoskeleton and adhesion between cell membranes. To mimic these 2 geometric constraints, we started from a simple experiment. We designed and 3D printed a 4 pentagons frame to represent the adhesion force between cell membranes and made a series of cell units with different materials. Notably, these cell units are primarily designed to be cylinders to imitate the expansion force from the cytoskeleton. We developed several generation cell units and found out that units with two ends in a round shape and a middle layer with a quadrilateral shape perform best. In addition, we found out as deformable material, silicon can smoothly perform the T1 transition, since this is the critical mechanism for generating scutoids and surface deformation. Combining the proper geometry design with the right material, we 3D printed, mold and cast several groups of the silicon cell units. The following experiment exhibits interesting results. As you can see from this video, when we assemble all silicon units together and bend the overall surface by hands towards the camera, the common boundary of two neighboring cells is getting longer. We then printed the frame with shape memory polymer and managed to get it bend on itself by providing environmental stimulation. Two silicon units were inserted in this bending frame. As the frame is bending towards the apical surface, you can see that the apical surfaces of two units are squeezing each other. The common boundary on its elevation is getting longer. In this video, 9 units were inserted in the frame and bending towards the apical surface. As the frame is bending, the common boundaries were getting longer and all units are performing T1 transitions. Moreover, we tested more units in a free-form surface frame and the results are still as expected. Through the process of developing the Scutoid parametric model and material study, we proposed that one of the necessary features of the scutoid is that either two vertices on the basal or apical surfaces merge into the center vertex on the intermediate layer. The connection between the three vertices creates a triangle that is partially bridging two epithelial cells. When 4 epithelial cells are packed as a cluster and bend toward the apical surface, the common boundary between the pair of adjacent hexagons becomes extended and the area of the connection triangle is larger. This mechanism creates an intriguing joinery method where two pairs of epithelial cells connect with each other in a bending environment. Greater curvature within the cell cluster also generates a larger area of triangular connection, thereby creating a more stable connection between two epithelial cells. The features mentioned above are well suited for application in masonry shell systems, as one of the main concerns when designing a masonry shell is to avoid sliding failure within the bricks. Compared with the simulation subdivision solution, the new algorithm that is used for designing scutoid embedded Masonry shell attempts to subdivide the overall surface in a rational and practical approach. The method creates more accessibility for fabrication as the subdivided components are unified into variable modules. This method aims to generate scutoid bricks for a static double-curved shell so that blocks can be interspersed with each other without the risk of sliding failure. Previously, we defined that the parametric model of a scutoid cell cluster can be recognized as a 3-layer structure. Two essential conditions for determining a scutoid are: 1. The number of the polygonal segments are not equal on apical and basal surfaces. 2. Two vertices on one surface can be merged into one vertex on the intermediate layer to equalize the polygon segments. Based on the above rules, we first extracted the 3 layers of a given shell geometry. Then, we subdivided the apical surface into hexagons and the intermediate layer into a variable grid of diamond shapes with hexagons, and basal surface into a grid of diamond shapes and octagons. Finally, we merged the 2 vertices of each hexagon on the apical surface with the vertex of each diamond shape on the intermediate layer and connected the same vertex of each diamond shape with the 2 vertices of the octagons on the basal surface. The above subdivision and connecting method create 2 types of scutoid bricks. One with the arrangement of hexagon diamond shape, and the other with the arrangement of hexagon diamond octagon. Despite the 2 different arrangements, both types of scutoids have 2 connection triangles, allowing these scutoids to be continuously assembled. In addition to the advantage of the connection triangles preventing sliding failures, we also discovered another benefit of the scutoid bricks in shell systems when making physical models. The connection triangles of both types of scutoid bricks, hexagon diamond shape, and hexagon diamond octagon, aligned with 2 bending directions U and V. as the given shell geometry is a doubly curved surface. The two types of scutoid bricks share a bridge of connection triangles along the respective axes. Meanwhile, the connected scutoid bricks on both directions act like multiple arches that are perpendicularly mortising together. The above configuration formed a space grid structure that is believed to have greater structural stability. Local The work that we have generated thus far contributes to and enhances our understanding of how deformation mechanisms within epithelial tissue structure local and global change due to changes in local geometry and global surface curvature. We also opened up a new channel that builds upon rigorous observations of cell behavior to then apply these findings in design research for architectural potential. Most importantly, we contributed a novel approach for shell structure design and fabrication from the perspective of individual scutoid units. These outcomes demonstrate that the epithelial cell-inspired brick can be applied in a masonry shell system and is a feasible and innovative approach for the fabrication of shell structures.